Sarah Davis. She's a PhD student in the College of Environmental and Life Sciences at the University of Rhode Island. She's going to be speaking to us about plastics today as she's studying plastic debris pollution in our coastal ecosystems. She's mentored by Dr. Colleen Suckling and Dr. Andrew Davies, and currently working on several projects focused on microplastics in Narragansett Bay, including seasonal surface trawl surveys establishing baseline population data, freshwater pollution surveys, and experimental trials investigating the movement of microplastics through coastal food webs. She's a former professional science educator, and she's particularly interested in engaging local communities in hands-on science research experiences, and she founded the community science program Mapping Microplastics during her previous master's degree work with the Miami University and the Wildlife Conservation Society. So I am very excited to introduce her tonight, and I'm going to get her PowerPoint queued up.
Sure. Thank you. That was everything. I don't need to give a talk now. Oh, cool. You're all set to go. Thank you. Thank you very much, Alicia. Thank you all for having me. My name is Sarah Davis, as Alicia mentioned, and I'm a graduate student at URI in Biological and Environmental Sciences. And as mentioned, I work with... Is it not working? I'm not sure how to make it advance to the next slide, but it's on this one. It's too fancy. There we go. Amazing. Okay. And as mentioned, I work with Dr. Colleen Suckling of the Fisheries, Animal, and Veterinary Sciences Department, and Dr. Andrew Davies of the Biology and Biological Oceanography departments. And our plastics team is called AUSIMAP, which is the Ocean State Initiative for Marine Plastics. And we're engaged in microplastics research and outreach in aquatic ecosystems here in Rhode Island. We're also really excited to be a part of the Plastics Land to Sea CoLab, that logo down at the bottom, which is URI's signature research initiative to better understand the plastics crisis through some interdisciplinary efforts. So I'm very excited to be here today to talk about plastic debris pollution, and I'll be touching briefly on the sources and impacts of microplastics, our team's research goals and some preliminary outcomes, and ways to be involved in research, advocacy, and actions here in the beautiful Salt Ponds community to keep our ecosystems protected. So, plastic debris, and especially plastic debris that ends up in our waterways, has come into really sharp focus in environmental science in recent years. Plastic is ubiquitous. We use it every day in many forms, and if we take a look around us at this moment or think about the places we spend most time in, it's impossible to be separated from plastic completely. And the fact that we use plastic is no surprise. It's very cheap to produce, it's durable, it can be manufactured into endless forms, and it has advanced technology a lot in most manufacturing fields. I like to view plastic itself as a neutral entity. It's a tool, just like anything. The life cycle of plastic, however, particularly end-of-life disposal, is where the problem occurs, and plastic really begins to shift from a neutral to a negative in the environment. And a massive amount of plastic waste never actually enters waste systems, and which really should be the first step in proper disposal. And as the global sink for most human-made waste, it's estimated that 13 million tons of plastic enter our oceans every year. We also may have heard the recent estimates that less than 10% of plastic is actually recycled when it does make its way into our global waste systems. Also, our waste systems really just aren't effective at handling really small plastic debris. So we can categorize plastic pollution really into two main size ranges. We have macroplastics, which are larger than 5 millimeters, so half a centimeter. And then we have microplastics, which are smaller than 5 millimeters in size, and they can go down all the way to 1 micron or 1 micrometer. And smaller than that, they become nanoplastics, which are very small. So from a marine perspective, there are some main sources we can point to of microplastic waste becoming pollution as it enters our waterways. So sewage-related debris, that number one there, is a major source of microplastic fibers from clothing and microbeads from cosmetics. much of our clothing is made of plastic. Polyester is one of the main woven plastic materials that we use as a textile, and every time we wash our clothes, we actually release thousands of fibers into the wastewater that flows out of our washing machines. And if we are attached to local sewer systems, most wastewater treatment plants can't filter out these tiny fibers, and they're just released at outflow points into our local waterways. And litter, think about some other areas, litter that's dropped in our towns and our beaches, just general mismanagement of trash is another main source. That stuff quickly breaks down in seawater and in sunlight into smaller fragments. Industry waste, such as from plastic refineries, as well as the shipping of virgin plastic materials from one place to another, they lose a lot in transit of those tiny little beads we call nurdles, which is how virgin plastic is first produced. And marine industries and recreation present a big challenge for plastic debris as well. Lost or abandoned equipment, such as nets and ropes, which are made of bundled plastic fibers, such as polypropylene or nylon, break down very quickly into tiny plastic fragments as well. Cut or abandoned fishing line might also be a familiar plastic danger to wildlife. So why are these plastic pieces a problem? Are they just inert when they enter seawater? We're really learning more and more every day about the impacts of microplastics in different aquatic environments, and we can make some general assumptions about the consequences of microplastic debris based on some evidence from previous and ongoing research. And in preparing to think about these impacts, it's important to recognize that the term plastic is really an umbrella. There are many different types of plastic used to make different items, and each of these polymers with their additives may behave differently in different ecosystems and present unique challenges. Additionally, they can be different shapes, contain different stuff, which are factors that could affect things like how easily they're ingested, for example. And one of the major differences between different types of plastic is their densities. And this is important for our research. Some plastics will float in water, up at the top of the water column, and some will sink. This means that different niches of ecological habitats, right, might be impacted by different plastics differently. Organisms that live down in the sediment, like clams, might be more impacted by things like nylon or polyester that are sinking to the bottom, whereas seabirds and fish who are hanging up towards the top of the water column might be more impacted by polyethylene or polypropylene fragments. This is, again, a really important part of our research process, which I'll touch on in a moment. So what are the actual impacts? Our main concern is ingestion, so eating plastics directly. And then we have what we call uptake, which is them entering an organism in some other fashion, mainly through things like respiration through the gills when aquatic organisms are breathing. It's well documented now that ingestion of microplastics can disrupt hormone regulation in many different aquatic species, and it can also create a false sense of fullness. So they feel full, but that results in nutrition deficiencies because they're not actually eating any nutritional food. And it can disrupt their digestive processes. So in general, it's clear that across the hundreds of studies done on different species, microplastics are just in general a major stressor. They decrease an organism's ability to carry out its essential functions, and it makes them more susceptible to other stressors they might be experiencing, like extreme temperature and disease. So another concern is microplastics as vectors. Like macroplastics, so like a plastic bottle, for example, microplastics don't stay in the area that they're first polluted in. They can travel huge distances and bring with them other stuff, including, for example, invasive species. In 2011, the tsunamis that resulted from the devastating earthquake in Japan resulted in plastic debris washing up in Hawaii that had Japanese invasive species on them. Marine organisms are adaptable, and they can find their home on floating plastic debris if they want to. So in addition to invasive species, they can also be vectors for disease. So most things, we probably know, dropped into aquatic ecosystems, whether freshwater areas or marine areas, they're going to biofoul or get that slimy layer of living microorganisms on them. And studies have shown that different microorganisms will colonize plastic that's dropped into the same area of seawater as wood fragments, for example, which is a little tricky because it means that we might have pathogens like viruses and bacteria that are traveling new routes on microplastics that they wouldn't otherwise travel. That could be an environmental and a human health concern. And lastly, microplastics can be vectors for other pollutants as well. So here are some examples of what we call the toxic cocktail of plastic pollutants, so things that plastics soak up really regularly, things like POPs, persistent organic pollutants, PCBs, hydrocarbons, and pesticides. So I know that this information can feel very overwhelming. It's a lot to take in, but the good news is that we have a very active research community in Rhode Island working to understand the current state of microplastic pollution in our areas and how we can continue to protect the landscapes that we rely on. So I'll pivot now to share some of our team's research and what we've learned so far. So currently, as mentioned, we have three main projects in the works. The first is a seasonal assessment of surface water debris in Narragansett Bay. We call this our man-to-trawl surveys. Next, we have a project assessing our state's freshwater resources, particularly how pollutants might transport through freshwater areas. And lastly, a project that's building on our understanding of the interactions between microplastics and organisms through trophic transfer, so predation, eating throughout the food chain. And that's with some experimentation at the mesocosm facilities at URI's Bay campus. So I'll detail each of these just a little bit. So I'll start by jumping into man-to-trawl. So as you can see here on the map of Narragansett Bay, we have 6 sampling sites that are spaced throughout the bay, and we have been sampling them seasonally for 2 years. We actually just finished our very last trawl today, which is very exciting. So this one was much more pleasant than our winter trawls, which are in February when it's 20 degrees, and we're spraying water all over the boat. So I'm glad to be done, but we've learned a lot, which is exciting. So we want to produce a really robust, technically sound approximation of what's happening in the bay, which is why our sites are spaced the way they are. We start up by Providence, so really urbanized, kind of urban to rural gradient, which is great. Not only adjacent to different levels of urbanization, but also different influences from things like currents, wind, ship traffic, rainfall, runoff, all things that might influence the spread of microplastics. So we're really hoping to just build a robust picture of what's going on, where these plastic particles are, and what they look like, what they're characterized of. So how do we do this? Well, you might notice we have some very interesting outputs on, which I'll mention in a moment. So we have a 330 micron pore size net, so it's basically a plankton net, and the holes on it are about 1/3 of a millimeter in size. And we trawl it behind a URI vessel for 10 minutes at each of our sites. It's called a man-to-trawl because, as you can see in the center, it has these wings on it, on its stainless steel frame that look like a manta ray. We call him Manta Claus, which I think is kind of fun. So all the debris gets directed into the cod end of the net. We have a water pump on board that we use to take in seawater, filter it, and use that to wash down the net in between replicants and wash everything into our sampling jars. So we have really robust contamination protocols on board and in the lab. This is because, just like the slide we looked at before, which showed how many fibers are shed from washing our clothes, we actually shed clothing fibers all the time into the air around us. And it would be really easy for us to mistake fibers from our clothing in our environmental sample. And we don't want to misattribute anything that we're looking at and skew our data. So our contamination color is orange. So we wear a lot of orange. We have orange jumpsuits, as you can see here, that we wear when we do field work that are very beautiful. They make us look like escaped convicts out on Narragansett Bay. And we have orange lab coats and masks in the lab. So this helps us identify anything that came from our clothing much easier, and we can remove it from our data, which is super helpful. So we also use filtered seawater on board to wash down the net, wash and putting into our sampling jars. So once we get back to the lab, we sieve it down, so we remove all the seawater. And then we actually add a hypersaline solution, so it's just super salty, saturated salt water, which ups the density a little bit and encourages any plastics that might be there to float. And then myself and our wonderful undergraduate assistants sit under the microscope and we hand pick out all the suspected microplastics. We don't just do this randomly, we use a protocol based on the literature, things like texture, color, density, shape. And we stick all of our suspected plastics to slides and capture a high-resolution image of them. So now we have all these things we think might be plastic, and then our last steps are really our plastic confirmation or identification steps. So the first one is very sophisticated. We poke them with a very hot needle to see if they melt. Plastics tend to melt at very high temperatures. Our needle is 320 degrees C. where lots of other materials will just burn or combust. So this is a way for us to really quickly rule things out. So we hot needle test every particle, and we've characterized over 3,000 particles from the bay so far, so it's a lot of poking. And last, we have our identification technique, which is called Raman spectroscopy. Raman spectroscopy is very cool. It allows us to actually identify the polymer type of the plastic that we're looking at. And it's what's known as a vibrational spectroscopy technique. So it uses a laser. We shine a laser basically at these tiny little plastic particles. And that laser light either comes back in the same wavelength as it started at. So if it started, for example, as green, it'll come back to the machine as green. But it also excites vibrations within the molecular bonds of the material. And what happens when it does that is that it sends back the photons from the laser in different wavelengths. And the machine reads that, and it spits out what we call a spectrum, or a spectrograph, up there in the top right-hand corner, that has these peaks at different wavelengths and at different intensities. That's basically a signature, a fingerprint of that material. So we can match that against a known database of different spectra, and we can have confirmation of what our plastic is. So we can say this is styrofoam, this is polyethylene, this is polypropylene, which is pretty cool. So results, what have we found? So this is very preliminary, as we are still processing a lot of our data. You can see it's kind of a complex lab process. But this is kind of what we found so far. So in this bar graph, we have, let's see if I can see my mouse in there, probably not, a north to south gradient from our site. So starting at CPO, that's Connecticut Point offshore, we get all the way to Beavertail Lighthouse down at the bottom. So we're going north to south here. And our different bars and different colors are different seasons. So we don't have any really strong seasonal patterns emerging yet, which is interesting. We thought maybe there would be more in the summer or fall as people are recreating more on the bay. But it actually seems like we might have some more in the winter. Maybe there's less disturbance in the winter, so we're able to capture more of the plastics, all just theories. But we do tend to see, as we kind of suspected, that we're recovering more particles generally in the more northern parts of the bay, up towards Providence. There seems to be, across microplastics research, a pretty strong relationship between urbanization and the number of plastics recovered. And again, we still have more samples to process, and this is preliminary, so all interesting to see, and again, it's just a picture of the sites from their north to south, kind of where they're located if you're interested. So comparing some outcomes, so we can take kind of an average concentration of how many plastics we're finding in the bay from these samples. And so up here in the right-hand corner is us, that's our Narragansett Bay average concentration of what we've processed so far. And we report this in meters cubed of seawater, so a cubic meter. And we found from our trawls about 0.3 microplastics per cubic meter. So you would need to swim through or trawl through about 3 to 4 cubic meters of seawater to get 1 microplastic. I think that's pretty good. And it also is pretty good when we compare it to some other places. If we look over to the left, to the Monterey Bay National Marine Sanctuary in 2021, using manta trawl methods, they found an average concentration of 1.32 microplastics per cubic meter. So we're definitely sitting lower than that, but we're also sitting a little bit higher from some other places. The Chesapeake Bay found 0.16. So we're kind of in the middle. A problem with comparisons, however, is that there's not a really rigid standard of methodology. So we have different methodologies, and those small variations could result in different data that we're getting. And one of those differences, which I'll get to in a moment, is the time of the tidal cycle during sampling, which is pretty interesting. But what do the plastics look like that we're pulling out of the bay? Happy to show you some photos. So we find mostly fragments, some beads, and some fibers. So beads generally look like this, plastic microbeads. Not too exciting. Fortunately, in 2015, the microbead ban was passed in the US. However, we're still getting microbeads that were polluted long before then in our samples, most likely. Fragments generally look like this. They might not have a really clear, distinct shape. They can look like anything, possibly from plastic bags, consumer products, lots of different things. Fibers look like fibers. We generally get fibers made from bundled polypropylene, Those nylon and polyester fibers, those are sinking to the bottom. So if we remember, we're sampling the top of the water. So we're getting that floating stuff, which means we know, we can predict kind of what kind of polymers we're getting from these pretty well. Colors, we get a lot of clear and white. Those are our main two, followed by blue, black, green, pink or red, yellow, and then tan or brown. And then polymer type, again, mostly polypropylene and polyethylene, and some polystyrene, which is styrofoam, as well as a good chunk of other that we're just unable to identify. So Raman spectroscopy, while cool, is not perfect. But we're still learning. So we were interested in kind of identifying the tidal variable in the number of microplastics recovered. So we did a couple of trawls that we call high-frequency trawl sampling, which is we picked 2 sites in the east and west passage of the bay, kind of on either side of Jamestown, and we trawled 10 times across one whole tidal cycle. So starting at high tide and trawling 10 times intermittently to the following high tide. So it's a long day out of the boat, a lot of trawling. But what we found was actually really interesting, and we are likely going to continue this tidal sampling to see if it's something that's replicated if we do it again. So we found that there's a strong relationship between tidal height and the number of plastics recovered. So these bars here are each of those 10 trawls, so from 1 to 10, and the height of the bar is the number of plastics recovered averaged. That line going through the middle that slopes down and the back up is tidal height. So we can see that as the tidal height goes down, as we approach low tide, we're getting way more plastics than as we're approaching high tide. So we have some theories as to why this might be happening. Just generally lower water volume. If there's less water and the plastics aren't moving out with the water, we're going to get more of them. Also could be as high tide goes out onto the beaches and shorelines, it's pulling more plastics back with it. All theories, preliminary. It's difficult to isolate all those variables with all the things that are happening in the bay. In the East Passage, we had a similarly strong relationship. We had kind of a peak out here happening, throwing off the data a little bit. But in the East Passage of the bay, we have much more influence coming from the open ocean down by Aquidneck Island, which could be influencing that kind of perfect tidal conditions. So this is interesting, and also is one reason why it's hard to compare between sites unless everyone is doing exactly the same thing, which there is a big push to do that, which is great. So we'll touch quickly on 2 other projects that we have ongoing right now. This second project is pump filter system sampling. So we've been sampling several freshwater areas with the Rhode Island Water Resources Council to kind of just get a baseline assessment of what's happening in our freshwater areas. And we've been sampling the Blackstone River a few sites, a couple sites in Roger Williams Park, Warden Pond, Flat River Reservoir, and Belleville Pond. And this is our pump filter system. So it's a little more stealthy than our Mantitrol net. We can take it right up to the shoreline, and we use an intake hose to just take in usually about 100 gallons of water per sample. And it flows, it basically comes in here. Walk over here. Comes in through our hose, and it flows across these 3 kind of canisters. Inside each of those canisters is a stainless steel cartridge filter with different sizes. So it starts at the largest filter, which is 280 microns, to 100 microns to 10 microns. So we can sample down to 10 microns in size, which is very small. And here we are again, we're in our beautiful orange in the middle of Roger Williams Park. Not being a spectacle at all. So we are excited to begin processing these. We're almost done with field work. We have a couple more sites. We have been trying to work with the Stormwater Innovation Center in Roger Williams Park to have a better understanding of how their stormwater infrastructure is affecting the transport of microplastics when they're entering versus leaving the park. However, it has not rained, so we are still waiting for more stormwater to be able to do that sampling. But we're excited to see what we find. The sample processing, I won't go into the details of this too much, is a little more robust. Oops. We get a lot of biological material, so we give it a chemical digestion to try to get rid of a lot of that, debulk it a little bit. We use density separation to get rid of sediment at the bottom. We can run it off the bottom of our samples. And then we filter our final sample onto filter papers, and we stain them, which is a pretty cool step, to use fluorescence to better identify plastic fragments. So we use Nile red to stain plastics and create fluorescence from that, and 2 stains called cauliflower white and Evans blue to stain biological materials so we can rule it out. So then we take a high resolution image of our filter papers. You can see here that blue fluorescence from our biological material, and we basically tag suspected particles on the computer and then take it to the Raman. So we have to use the Raman on every suspected particle to try to see what we found. And this is just some cool visuals of fluorescence. So this is under normal light conditions. We have some beads here just for visual. These are beads that we put into the sample. And then under fluorescent conditions, we can see that blue biological material glowing really strongly so we can rule it out as being not plastic. Our beads are glowing super brightly, and we have a suspected plastic there glowing in the middle that we otherwise might have missed if we were not using fluorescence, which is pretty neat. So our last project that we have in the works is our trophic transfer experiments. So we are conducting these in URI's mesocosms, which are a really neat facility. There are 10 in-ground tanks that are right next to Narragansett Bay. And these tanks use flow-through seawater, so we can create basically a mini ecosystem of Narragansett Bay right next to the real one. But in that mini ecosystem, we can manipulate things we otherwise would not be able to. For example, we can add plastics, which if we did that in Narragansett Bay would just be littering, and ask some interesting questions. So we're particularly interested in looking at blue crabs and how they might be acquiring plastics in their body through the ingestion of other species, oysters. So we had a master's student, one of my colleagues last year, do her work on the intake of polyester fibers by oysters, and so now we're taking that a step further in the food chain and adding in blue crabs. So kind of seeing how that process might be working. This picture is of one of our undergraduate interns, Ashley. She just finished up a small project this summer looking just at blue crabs and ingestion of microplastics, and she was really interested in how that impacted their fecal matter. So we spent 10 days collecting poop from blue crabs, which if you have never done that, collected poop from an angry blue crab, it is very character building. I highly recommend it. So we're excited to get those going in the big mesocosms next summer. So how can you be involved? We're really interested in engaging our local community in some different types of research experiences. Let me just find my correct page here so I can tell you all the great things. Okay, so we really have 3 main ways of involvement. The first is what we call grab sample collection. So this is something we've been piloting with Watershed Watch this summer, which has been very exciting. So it's accompanying our freshwater work currently, but we're interested in expanding it to other sites. This is pretty simple. It's just scooping up a water sample in a glass jar and recording the date, time, and location, and we take that back to the lab and process it. So similar to what happens with, it sounds like, the pond watch samples as well. So it gives us like a snapshot of what's happening at that time in that sample of water from a plastics perspective. The second is by helping us identify litter that we've collected from the shoreline of our freshwater sites. So this is something that one of our undergraduate research interns has been conducting as a small project, and we've collected lots of litter from plastic shorelines, and we use the United Nations Environment Program means of categorization. So we just are looking for help categorizing all the litter we found into their codes that they designate things at. So we use that doing just online surveys. So if that's something you're interested in, we'd be happy to get more folks involved. And the last is some community science or citizen science microplastic sampling. So this is related to the project that I developed in my master's work, which is called Mapping Microplastics. It's a little more involved than taking grab samples. This is something that folks will process and record the data on their own, which is pretty exciting, makes it a really good classroom activity, things to do if you have a really eager middle schooler who wants to be involved in science, things like that. We're really interested in using this as a tool not only to record data about microplastics, but also to engage our local community in some hands-on science and some local nature experiences as well. So how does this one work? Folks will collect a water sample. They'll filter it, so we use just a coffee filter stacked over a glass jar, so we're just getting the debris out of the water. And then we actually use Nile red stain, so it's the same thing we use in our lab to create fluorescence. We're looking for larger microplastic pieces in this one, about 1 millimeter or larger. And then we use blue lights and yellow glasses to view the fluorescence. And so folks just record, take a picture of what they see on their filter paper, send it in, and we add it to an online map so we can see what other citizen scientists are finding. We're also right now building, working on 3D printing, a tool to make this kind of more all-in-one, which would be really neat to kind of simplify the user process for folks who want to get involved in this. So if this sounds like something that you'd be interested in getting involved in, I can share my contact information and I can keep you updated on when we kick things off. So aside from research, what can we do as individual residents of Rhode Island to help protect our local ecosystems from this plastic debris? While we wish there was a panacea, positive impact really can come from individual and community actions to prevent new plastic from entering the waterways, and participating in cleanup efforts to mitigate the current problem is also really helpful. And like most environmental issues, the responsibility, impacts, and future of the plastics crisis is really deeply embedded in issues of social justice, equity, and access. And individual actions might seem small, but if they're accessible to you, they can be hugely impactful as they accumulate large numbers of engaged and informed community members. And we've probably heard of some simple actions to reduce our plastic footprint, so to speak, such as choosing alternatives to single-use plastics, like skipping the straw or bringing reusable bags to the grocery store. COVID did set back our ability to choose alternatives to single-use plastics in many fast casual dining or coffee shops, but we're hoping that begins to kind of shift back as we enter this new post-COVID reality. And as folks who live near the salt ponds, you're actually in a unique position as there are some actions you can take as recreational users of these areas to help prevent microplastic pollution as well. So if you have a boat or regularly tie up things like canoes or kayaks, you could consider replacing old or fraying marine ropes, which are likely made from plastic materials like polypropylene or nylon, with ropes made from natural materials like sisal or hemp or manila. This is a choice each user has to navigate based on how much they use their recreational boating equipment and kind of what you use it for, as there's benefits to using plastic-based lines, but they shed plastics almost constantly. And switching to natural materials can be a huge help. Another useful swap is to choose to wear clothing made from natural materials when actively recreating on the water, such as cotton or linen or wool. This might not always be an accessible alternative depending on weather, since clothing with waterproof lining is usually plastic-based, but it can be a conscious choice to make on fair weather days. For those who use vinyl or PVC-coated stainless steel traps like lobster traps or crab traps, a good practice that will not only help your traps from shedding those plastic linings, but that will also lengthen their lifespan, is to always transport them really mindfully. So never drag them across pavement or on docks, because that lining will come off really quickly. You can also, if you only set them out for a short amount of time or use them just seasonally, you can also skip the anti-fouling paint. That paint chips off super fast and you have to replace it every season. And it's just as effective to pull them out of the water and give them a freshwater rinse when you're done to prevent that algal and microbial growth. Oops. You can also replace old styrofoam buoys with buoys made from a hard plastic. It's a better long-term choice because styrofoam falls apart, as we know, really quickly in water and is really hard to recover. And when winterizing your boat, you might choose a longer-lasting alternative to shrink wrap, such as a reusable boat tarp. And while many reusable boat tarps might still be made of a plastic material, they don't need to be disposed of every spring and can be used for many winter seasons. A quick Google search will yield many recommendations on different materials and instructions for switching from shrink wrap to reusable tarp. And if you do shrink wrap your boat, Woods Hole actually offers a recycling program every summer and has yielded like thousands of tons of shrink wrap recycling, which is very cool for kind of our New England area. So while this is no means an exhaustive list of actions, I hope it offers some ideas of ways to incorporate plastic mindfulness into your habits around the salt pond areas and provides a look into the world of microplastics research here in Rhode Island. Our team is looking forward to continuing our ongoing projects and sharing the results with our community over the next couple years. And with that, I'll say thank you so much to our supporters, to Alicia, and to all the Salt Pond Coalition's members. I'm happy to take questions if there's time. I know I spoke for a while, but thank you very much.